Okay, could somebody please confirm that you're hearing me?
Yes.
Yes.
Great. Lab. Oh, that's the wrong. Let's share the screen. My computer's a bit slow because converting the meeting. Sure. Let's scroll off of that. All right, are there any questions? Okay, great. So, oops, no, we're supposed to be there. We're supposed to be here. We're doing lab 2 aseptic transfers today. Any questions about what we're doing? Remember, you need to read the textbook in chapter 3 about the section dealing with compound light microscopy. That needs to be read before beginning labs 3 and 4. If there's no questions, I'm going to close that down because my computer is a little overworked at the moment. That'll happen until the lesson plan finalizes. So lab 2, module 2, aseptic transfers. The learning objectives for this lab is you should be able to understand the purpose of using aseptic transfer techniques, determine if aseptic transfer was used from the data that's provided. So we'll give you some data and then you tell me, was successful aseptic transfer used or was it unsuccessfully used? And then define the following terms. What's ubiquitous, aseptic transfer, inoculation, and a contaminant. There are some videos in today's lab, and they're demonstrating a procedure in the lab. In our local environment, you should know that microorganisms are ubiquitous, meaning that they're essentially everywhere. This is problematic when working in a microbiology lab because we generally want to work with pure cultures, a culture that contains one and only one species of microorganism. The technique for getting a pure culture was developed in the mid-1800s by Louis Pasteur, and he also developed the technique for making a aseptic transfer to minimize the risk of culture contamination. Now, how he did that was actually very difficult. He had to take his bacteria culture and then dilute it at certain densities and then test the density. And if in that density, 1 tube out of 10 tubes had growth in it, and then 9 tubes had no growth in it, he correctly concluded that he had a good chance of having a pure culture in the 1 tube that had growth and 9 tubes of the same density had no growth. And he had to do a lot of work. He had to make the dilutions, and then he had to make many different dilutions, and then make replicas of that dilution, and then put it in a tube, or I guess that would be the replica. And you'd have 10 of each dilution. And usually he did at least 5 or 6 different dilutions. Sometimes he did 10 or 12 different dilutions and 10 tubes each. That's a lot of tubes he set up. And all he got at the end was 1 pure culture. My point is, it was a lot of work and it wasn't easy for Louis Pasteur to get a pure culture. we will talk about an easier way of getting the pure culture that Dr. Koch invented. We're going to learn how to make an aseptic transfer in this lab. So during inoculation, never lay your test tube cap down on the workbench, which I don't think I can take this off, but I can go like that. I don't know if you can see that. Never lay it on the workbench. Always hold on to it. Having it in, this is a test tube, obviously pretend it's a test tube, and we take off the cap. You should hold the cap in your hand and then hold the test tube either in the same hand or a different hand. And best thing to do is to hold it like this and then get a loop, sterilize the loop, and this is your tube. We'll go like that. And then get the bacteria on the loop and then add the bacteria to the tube and then put the cap on. So never lay your test tube cap or petri dish lid on the workbench because it can become contaminated. To minimize contamination when inoculating media, tilt an uncapped agar tube and hold the petri dish lid at an angle. When you have the tube held at an angle like this, if bacteria from the air come down here, it's not going to go in the tube, it's going to go outside the tube. And so when you hold it at an angle, you have less risk of something from the air coming down and falling in the tube. Any question about any of that? And then the same thing with the lid. When you're working in a petri dish, you should have the lid partially over the petri dish so that if something comes down, most of the time it's going to hit the top of the lid and won't go in the petri dish. To go in the petri dish, it has to fall right down here. And you do have to have it open when you're working, so you have to have a little bit of risk. But there are things to reduce the risk, and these are two. Another major thing for reducing the risk is have the lid open on the tube or the petri dish for as little time as possible. And that will reduce the airborne contamination falling into the dish or the tube. And that was one of your questions in a previous lab. Making aseptic transfer. In order to inoculate, transfer cells into a new sterile media, we must make sure that we sterilize everything that can potentially contaminate the sterile media, meaning the media and the tube that you're giving in a microbiology lab will be sterilized for you. Usually they will be sterilized in the autoclave. And then when you're working, you're going to use tools to sterilize your tools that you're working with. Like you'll be working with a loop. Let me save this down, then my computer will now work at a better rate. Like when you're working with a loop, the one looks like this, where it has a wire that ends in a loop. And this is good for picking up cells in this loop. And then this is a metal portion of the loop. And then this is the plastic or rubber portion, so that when you heat up the loop, it won't burn your hand with the plastic or rubber portion. And you can sterilize this loop by placing it in the flame of a Bunsen burner, which we no longer use in the microbiology lab. In our lab, we use a Bacto incinerator, and you turn it on, and the stone inside, that white part right there is stone, it will become red hot, and you put your loop inside the Bechtel incinerator until the loop becomes red hot. And that sterilizes this portion of the loop. You do not sterilize this portion of the loop. Obviously, that would melt the handle, and you don't get this part even red hot because that would melt the handle as well. But you do get this portion red hot. Any question about that? Let's just sterilize it so that you don't have microorganisms on this loop. And then you cool it. And then you put the loop in your stock culture and then transfer from your stock culture to your sterile medium. So know the overview of the terms. I'm not going to go through them, but you can read through it. And then you do laboratory 2. You're not physically going to be performing any of these activities, but you will be watching videos of these activities. And we expect that you will know and understand the material in these videos. In procedures 1 and 2, in procedure 1, you're going to be transferring sterile liquid water using a glass serological pipette and putting it into media. And in procedure 2, you're going to transfer a sterile paper disc using sterilized forceps to put the paper disc into a tube of liquid media. So watch the procedures in the video here demonstrating proper aseptic transfer technique. In procedure 1, as I said, you're going to transfer 1 ml of sterile water into nutrient broth. When you're transferring 1 ml, you should realize that water and many other things that when you're measuring on the pipette or the birette, that's not how you say that. Close enough. Colander, it's marked, birette. The water will form a meniscus where it'll be higher at the sides and lower in the center. How do you read this? Do you read it from the bottom of the meniscus, from the top of the meniscus, or the average? Anybody
The bottom
know?
of the meniscus. Oh, sorry.
Say again?
Oh, I said the bottom of the meniscus. I
Yeah,
didn't mean to
you
interrupt.
always
I'm sorry.
read from the bottom of the meniscus. And water will do that as well as many other substances. I don't think oil does, but most other substances do. Of course, most other substances are based on water, but in procedure 2, you're going to be using aseptic technique to transfer a sterile paper disc into the nutrient broth. All of the tubes and containers must be labeled with your initials, the date, what the media is, and then the temperature that you're growing it at can be optional. So once you transfer either in procedure 1, the liquid in, or procedure 2, the paper disc in, you then cap the tube and then put it in the test tube rack and put it in the incubator. And then a day later, you look at it. For procedures 3 and 4, you're going to be transferring Serratia marcescens. In procedure 3, you're going to be transferring Serratia marcescens into a broth. For procedure 4, you're going to be transferring Serratia marcescens to an auger slant. And we have a video demonstrating how to correctly transfer from a bacterial stock solution to a sterile broth and to a sterile agar slant. So watch the video here. Some notes about the video. You only need to watch the video to 4 minutes and 23 seconds. In the video, they're transferring E. coli, which is different from Serratia marcescens because E. coli is non-pigmented or an off-white color, meaning off-white is not really colorized. It's not a color, so it's not pigmented. Whereas Serratia marcescens is pigmented, it will be either reddish on a solid media or pinkish on a liquid media. There's an error made when the instructor puts the loop into the E. coli stock tube, and you can hear the error from 3 minutes and 5 seconds to 3 minutes and 20 seconds. You should look for that error. You'll hear a sizzling, and there may be a question about what is that error. Procedure 3 is using aseptic technique to transfer Serratia marcescens to sterile broth. With Serratia marcescens growing in a broth, it should look pink. Broth cultures are problematic because it's difficult to determine if contamination has occurred because all the cells will mix together in the broth. And so you won't see one colony of one type and another colony of a second type. They will all look similar because they're all mixed together. That's stated if you have a colored species that's growing in a broth and the color is correct, then you know that if there's contamination, it's only minor contamination because at least 90% of the cells are correct because it has the correct color. But if there was 10% contamination, you would not see it. And that's a problem when you're working with the broth. Minor contamination you wouldn't see. So for procedure 3, label the tube with your name, the date, the type of media, which will be NBYE, nutrient broth with yeast extract, with the incubation temperature, which is optional, and the inoculant, which would be S. marcescens. Put the bacteria in, cover the container with the tube, and the tube should look like that before you begin. And then take a look at it after overnight incubation in the incubator at 30 degrees Celsius. And then you look at the table here to see what the result is and the conclusion. And I'll come to that after talking about procedure 4. For procedure 4, it's very similar, except you're transferring Serratia marcescens onto a sterile agar slant. When Serratia marcescens grows on an agar slant, it'll be reddish in color. Contamination will be much more easily to detect because if the color anywhere is not red, then you know you have contamination. Let me come on over and show you a picture. So there you have the tube, and it's growing red. If there was any region, like a little area that's white, we know we have contamination, okay? And it's just much easier to determine contamination on a slant than a liquid culture, a broth, because on the slant, the contaminant will all grow together and it'll have a different color than what you're supposed to inoculate it with, the Serratia marsens. Any question about any of that? All right, so you put the tube in the incubator and then you take a look at the tube. For procedures 1 and 2, you use the result here. If the nutrient broth is clear, the conclusion is no organisms are growing in the broth, aseptic transfer was successful. If on the other hand, the nutrient broth is turbid, hazy or cloudy, organisms are growing in the broth and aseptic transfer was unsuccessful. For procedure 3, if the nutrient broth is clear, no organisms are growing in the broth and aseptic transfer was unsuccessful because we transferred Serratia marsens into the broth. If the nutrient broth is turbid, hazy, or cloudy, then organisms are growing in the broth and aseptic transfer is generally successful. You have to make sure the color is correct before you can say it is 100% successful, but at least organisms are growing. For procedure 4, if the slant does not show confluent or colony growth, no organisms are growing on the slant, so aseptic transfer was unsuccessful because we purposely put Serratia marsens on the slant. If, on the other hand, the slant does show confluent or colony growth, organisms are growing on the slant, and aseptic transfer was successful. For procedures 1, 2, 3, and 4, you're not going to be doing it. So we've done it for you and provided the pictures. Here is a picture of procedure 1 after a day of incubation. There is a picture of procedure 2 after incubation. You can actually see the paper disc at the bottom. This is for procedure 3 after incubation, and this is procedure 4 after incubation. Use these pictures to get your observations, your result, and then make your conclusions, the interpretation of the results. Use this table up here to get your conclusion, okay? And you should word your table, you should word your answer the way it's worded in the table, okay? All right, so then answer the questions of the worksheet. So let me show you the worksheet now. And it does, the worksheet does have the links for the videos. And then has the procedures. So answer the questions, the first question being 0. Describe the initial clarity of the nutrient broth prior to inoculating either the sterile water or the paper disc, and state whether it was clear or turbid. And then for 1, this goes to the result of procedure 1. Describe the clarity of the nutrient broth after inoculating with sterile water in overnight incubation at 35 degrees. Is it clear or is it turbid? And we have a picture right here. That's procedure 1. So answer that. Based on these results, what conclusion can be drawn? Use the table to answer the conclusion. And it should be worded similar to the wording in the table. And then list 3 sources of error with aseptic technique that could explain why the nutrient broth showed what it did, growth of microbes. So 3 possible errors. Okay, and they don't have to be necessarily the one that did it, but they have to be a possible error. For result 2, describe the clarity of the nutrient broth after inoculating with a sterile paper disc and overnight incubation at 35. Was the tube clear or turbid? Based on these results, what conclusion can be drawn? For procedure 3, answer question 3. The results for procedure 3 describe the clarity of the nutrient broth after inoculating with Serratia marsans in overnight incubation at 30 degrees Celsius. Was it clear or turbid? So your answer should be either clear or turbid, by the way. And then give your conclusion. Once again, use the table to write your conclusion. And then answer, how do you know whether you've transferred the correct microbe, which is Serratia marsans? And then for result 4, is there bacterial growth on the slant? Just answer yes or no. And how do you know? State how you know. And does it appear to be the bacterium inoculated, or does it appear to be a contaminant that's growing? And then that's it. Submit your answers, the worksheet, to the folder in Canvas for lab 2. I will be here until 8 o'clock or until the last student leaves. Generally, so far, no student has stayed to the end time. And generally in this lab, students tend to finish early. So if you have a question, I will be here to help you, but don't wait until the end in case nobody's on, in which case I'll log off. All right, go ahead and get started in the lab. I will be here to help you with the questions.
Thank you.
You're welcome.